Welcome to MycoTalks on behalf of myself and my co-host Jay Coles. We have two fantastic physician scientist speakers today, George Deepe from the University of Cincinnati and Tamara Doering from Washington University. You see the names of the organizing committee for MycoTalks listed below on the slide here. And I want to thank and acknowledge the MRC Center for Medical Mycology at the University of Exeter for organizing MycoTalks and bringing us together as a community. Just to remind ourselves of the philosophy and origins of MycoTalks, we promote diversity at all levels, career stage, gender, geography, and field. There are two 30-minute talks on all aspects of medical mycology and then 30 minutes of in-depth discussions. And just a reminder to please submit your questions through the Q&A function that we'll be able to see, and please include the name of the speaker. An especially attractive feature of MycoTalks is that recordings of the talks will then be available on the MRC YouTube channel. To complement MycoTalks, there's a corresponding series called MycoClinics. The next one is being held on March 5th about trauma-associated fungal infections. And the organizing committee is a group of illustrious medical mycologists with clinical focus. So feel welcome to participate in MycoClinics as well. So it gives me great pleasure to introduce our first seminar speaker, George Deepe from the University of Cincinnati. And George is really an exemplary physician scientist. He's an outstanding medical mycologist, and he's a terrific colleague. And he's going to be speaking to us today about zinc and host defenses to histoplasma. So George, please go ahead and turn on your camera.
Let me begin so I don't eat into Tamara's time. So I want to, instead of going over the pathogenesis of histo, because you've heard that from Chad Rappley and Anita Sill and others, I'd like to give you some fun facts about histoplasma. Number one, it was discovered by Samuel Darling in 1905, hence the term the darling of the Ohio River Valley. And I thank John Galgiani for that moniker. This organism, like other organisms, is moving around because of climate change and other changes. So the spores have heated Horace Greeley, who is an American journalist, urging, "Go west, young spore." It used to be "Go west, young man." Now it's "Go west, young spore." This has infected at least one Nobel laureate, and that is Bob Dylan. And Joe and I were talking, and I think Alfred Gilman and Michael Brown from UT Southwestern were also infected, but I don't have medical proof of that. So this can be a dangerous disease, especially for Bob Dylan. And finally, it is the only fungus that I'm aware of that is referred to in a song, and it's by Johnny Cash, called "Beans for Breakfast." And I think this is really apt for our British colleagues who love to have beans for breakfast. So if you're interested, you can go and listen to Johnny Cash's song. It's a great song. Before I begin talking about histo per se, I want to introduce you some of the tools of the trade that we use with our chemistry colleagues, and the measurement of zinc is done by inductively coupled plasma mass spectrometry. This is an analytic tool that is used to analyze elements, elemental determinations. It wasn't introduced until 1983, and it now is recognized or is used by many laboratories who want to measure metals. One of its advantages is it has high energy ionization source, up to 10,000 K, and is very efficient in ionizing a wide variety of samples. What happens is the sample is typically introduced into the nebulizer and is introduced as an aerosol. Once the sample is introduced, it gets desolvated, atomized, and ionized. And then the instrument can separate by mass spectrometry according to the mass charge ratio, which allows for identification and quantification of up to at least 30 different metals. So it's a powerful tool in terms of metallomics. So let me begin with the background information about how we got where we are. And I'm going to give you short vignettes of work over the last several years rather than a complete novel. So we had known for a few years that endogenous GM-CSF is essential for survival of mice infected with Histoplasma capsulatum. We published that years ago. We also had demonstrated that if you administered recombinant GM-CSF to mice, it enhanced the clearance of the fungus, particularly in immunosuppressed mice, that is T-cell depleted mice, when we used it in combination with a suboptimal dose of an antifungal, and that antifungal was amphotericin B. A colleague of mine, Simon Newman, who was principally interested in macrophages, had demonstrated, as did we when we followed up, that GM-CSF arms both mouse and human macrophages to inhibit the growth of Histoplasma capsulatum. What's missing is how did GM-CSF do this? What we focused on was nutrient availability to the intracellular yeast, and we focused on metals as kind of a point of initiation. And what we found was the following. When we infected macrophages, whether they were peritoneal or bone marrow macrophages, and I'm showing you the data with peritoneal macrophages, when we infected the macrophages with histoplasma, either in the activated state or the unactivated state, and then we measured a variety of metals through ICP-MS. And you can see from the panel on the left that macrophages that were resting and infected acquired more zinc, a little bit more iron, not much copper, and not much manganese. We measured several other metals, but I don't show you that data. If we activated the macrophage with GM-CSF, you can see you do get a wee bit more of zinc and iron. And if you activate them and infect them, there are increases in zinc, but not iron in the macrophage itself. We developed a technique to isolate the yeast and measure the total elements in the yeast. And as you can see from this figure, only zinc was limited. There was a decrease in zinc compared to resting macrophages down here. So what was happening and what we surmised from this was that GM-CSF was activating the macrophage and histo wasn't faring very well. And the reason was there was less zinc available, not less iron, not less copper, not less manganese. So this suggests that because you have an increase in total zinc in the macrophage and less zinc in the organism, there might be, or there is, a protein or some molecule that's sequestering zinc away from the yeast cells. To address this issue, we actually turn to size exclusion chromatography that is coupled to ICP-MS. And that way we could examine the distribution of zinc in the metalloproteome of both histo and the macrophage. And using size exclusion chromatography, we'll separate molecules by sizes, as I mentioned, and metals can be detected in individual fractions as they're collected. And what we found was the following, and I'll go over this. So this is an activated macrophage profile from the HPLC. And there are two spots to look at. One is this hump right here, and one is this small hump right here. So this represents when we did proteomics metallothioneins, particularly metallothioneins 1 and 2, and this represented the labile zinc portion. So this can be quantified by area under the curve. When we looked at the GM-CSF activated macrophages, what we saw was a marked increase in metallothioneins 1 and 2, and a concomitant decrement in the amount of labile or free zinc. And we could quantify that by fraction. What you see is a progressive decrease in the amount of labile zinc as you go from a resting macrophage to an activated macrophage to an activated macrophage infected with histo, and you see a concomitant increase in the amount of metallothioneins. So these results suggested to us that metallothioneins were binding zinc and depriving the organism of that metal. So what are metallothioneins? They're really small metalloproteins. They're omnipresent. They're in all organisms. They're multifunctional, highly involved in zinc, but not just zinc. They can bind cadmium, they can bind copper, they can bind a number of other divalent cations. And they're thought to have evolved to prevent heavy metal detoxification, especially something like cadmium, which can damage DNA. There are a number of inducers of metallothionein expression, and cytokines are just one of them. So we asked whether the absence of metallothioneins actually alters the clearance in macrophages that are stimulated with GM-CSF. So we got MT12 knockout macrophages, and when we cultured them and stimulated them with GM-CSF, what you can see is that in the wild type, there's a nice decrement in the number of colony-forming units, but this wasn't true in metallothionein 1 and 2 knockout macrophages. And this was accompanied by an increase in the total amount of zinc in the organisms recovered from the MT12 knockout macrophages as compared to the wild type. So the absence of MT12s, no sequestration of zinc, more zinc in the organism, the more it can withstand the onslaught from GM-CSF. So then we asked, well, are MTs and zinc vital in the generation of an ROS response? Now, it must be pointed out that ROS, histoplasma, actually induces ROS, and it is can withstand attack from ROS under certain conditions. But when we looked at MT12 knockout macrophages, first we looked at a low zinc environment. And when you look at a low zinc environment, you boost the amount of ROS substantially. And when you look at infected macrophages from an MT12 knockout, that ROS response is diminished. So how does the ROS response, how is it regulated in the context of GM-CSF activation? And the answer lies with a protein called hydrogen-gated voltage channel 1, otherwise known as HV1. And this is located in endosomes or phagosomes, and it is a protein channel. And it allows hydrogens to go into the phagosome in this particular case, and facilitates the acidic environment, which will enhance the activity of NADPH oxidase. GM-CSF activation of bone marrow of HV1 negative bone marrow macrophages did not reduce the burden of use. And I'll show you that here. So HV1 was very important in that process and in the generation of ROS. Now, let me go back. Now, as I mentioned, histo is kind of can withstand ROS attack, at least certain levels of ROS. And it can do so by superoxide dismutase production. And Chad Rapley had shown very nicely that superoxide dismutase, the zinc and copper one, which is extracellular, does not function in a copper independent environment. And we had found evidence that it does not function in a zinc independent environment. Therefore, what you lack is a mechanism to withstand the ROS when you have low zinc, accounting for the fact that histo may be more susceptible to ROS in the absence of zinc. So there are two things going on: more ROS because of less zinc, and more susceptible or more vulnerable to attack by ROS. And this just kind of shows you our schema. To go quickly through it, GM-CSF activates the macrophage. The macrophage MTs 1 and 2 go up. You get sequestration of zinc. The zinc then alters the activity of the hydrogen-gated Foltes channel, and therefore you get less zinc import to the fungal pathogen, less superoxide dismutase 3, presumably, and more vulnerability to the attack by ROS. And in fact, 10% of the proteome of histo requires zinc, and so there would be probably more other additional changes to its ability to grow. Now I'm going to switch to another story. This has to do with, again, with metallothianines, but a different twist. And in histoplasma, Th1 response is responsible for clearance. A Th2 response, principally by IL-4, is responsible for the progressive nature of the organism, and in the case of mice, the death. So we looked at IL-4 and whether it affects zinc just as a screen. And what we found was that when you incubated macrophages in the presence of histo or without histo, more zinc was accumulated by the macrophage, labile zinc by the macrophage, induced by IL-4. And we found this to be not only true in the macrophage, but also the histo was acquiring more zinc once inside the macrophage, and once inside an IL-4 activated macrophage. There was no more iron, no more copper, and no more manganese. The iron is somewhat surprising, given the fact that IL-4 is known to increase the transferrin receptor expression. But these are the data. So what we did then is, how is the zinc getting in? And we used a process called isotope enrichment model. And so the macrophage cultured in medium will have normal zinc. We can label yeast cells with 66 zinc, and we can label the media by removing all the old zinc and replacing it with zinc 68. So now you have an extracellular environment with zinc 68, enriched for zinc 68, the yeast with zinc 66, and we can look at the various isotopes of zinc in both the yeast and in the macrophage. So in the case of IL-4, what you would find is presumably more zinc 68 getting in, more zinc 68 be found in the yeast. That's the ideal situation. So what did we find? Indeed, when you look at IL-4 stimulated macrophages and look at the histoplasma, they were acquiring more zinc from the macrophage, which was acquiring more zinc from the extracellular environment. Therefore, IL-4 was actually enhancing the ingress of zinc into the macrophage, and that was ending up in the phagosome into histo. So what does it mean? We asked where this zinc could be coming from. So turn back to metallothianines. So we've already looked at metallothianines 1 and 2, and those are known to bind zinc. Metallothianine 3, though, is different. It is a zinc donor. And so we did a real-time PCR looking for the induction of metallothioneins in the presence of IL-4, and we found that both IL-4 and IL-13 induced a upregulation in MT3, but not in MT1 and 2. And we looked at several other cytokines which did not modulate metallothionein expression. Now we also put together a known fact, that is IL-4 is a known stimulus for the generation of cathepsins. And since zinc levels were elevated in IL-4 stimulated macrophages and yeast cells, we asked if this was caused by cathepsin activity induced by IL-4. So here again is part of a SEC ICP-MS. And what we did is we incubated the macrophages with inhibitors of cathepsin activity, pepstatin A and ALLM, and looked at the labile zinc quantity. And you can see that in the untreated macrophages is high. This is IL-4 treated. When we inhibited with the cathepsin inhibitors and we used the combination, there's less labile zinc, suggesting that that cathepsin activity was important to production of this labile zinc quantity. And when we quantified it, you can see that zinc in the presence of inhibitors, there was less zinc available. So again, indicating that cathepsins were being turned on by IL-4. These were cleaving the metallothioneins to release zinc. And this is illustrated in this graphic where IL-4 comes and engages its IL-4 receptor. It's through STAT6 and the JAKs. You get STAT6 transcription activity, IRF4 leading to an increase in metallothionein 3 and some zinc transporters. This metallothionein 3 transcription leads to the production of MT3 protein with bound to zinc. In the lysosome or phagosome, MT3 is there. In the presence of cathepsins, it causes the MT3s to release zinc, and that then can get into the phagosome where the yeast can take up the zinc and be fortified from being killed. So that's story number 2. Story number 3 has to do now with the organism, which is not much to do. Our lab really doesn't do as much with the organism as we should, but we, thanks to Logan Blanchett, we created a reporter strain for the zinc importer that we identified as ZERT2. And previous work by grad student Jessica Day had shown that if you knocked out this gene in histo, it didn't fare very well in the organism, particularly during the adaptive phase, indicating that ZERT2 was important for survival. So we created this ZERT2 reporter using GFP for the ZERT2, and then to identify infected yeast, we used D-tomato, courtesy of Chad Rapoli. So what you would get is, if you're in a normal or high zinc environment, you would see the organism by red, and the GFP would be low. In a low zinc environment, you would expect to be more GFP and maybe less red in general. So first of all, we tested it out, whether this reporter actually works. So we incubated it in the presence of low zinc quantities or in the presence of the zinc chelator T-pen. And you can see if we just looked at by flow cytometry, the MFI, the ZERT2 was turned on quite a bit. And if we did it with GM-CSF, we found the same thing, suggesting that GM-CSF-induced macrophages turned on the gene. So the organism was sensing a low zinc environment. And again, this reproduces that data, showing you that in the presence of GM-CSF, the MFI of ZERT2 goes up. If you add back zinc, it goes down. And it wasn't true for a control. We used interferon, and it wasn't true for interferon. Now, when we looked in vivo, we fractionated the macrophages into total macrophages, in interstitial macrophages and what we call exudative macrophages. And the reason why we did it in another study, but the reason why we didn't do alveolars is because we're using GM-CSF knockout mice and they don't have alveolars. And you can see that contrary to what you might expect, we didn't see a decrement like we did in vivo in each of the macrophage populations that we looked at. So this was puzzling. So we asked, well, maybe MCSF was important. So what we did is we looked at, first of all, whether MCSF influences host control of infection. And it doesn't in an intact mouse, but it does cause a slightly more aggressive infection in a mouse lacking GM-CSF. So suggesting that MCSF does contribute only when GM-CSF is present or absent. And then we looked at the zinc reporter and showing that when you neutralized MCSF, so you had higher burden, you had less zinc reporter activity, suggesting that histo was not sensing a zinc depleted environment as much in the presence of when MCSF is absent. Therefore, the absence of both GM-CSF and MCSF are required to really enhance the expression of this zinc transporter. In my last story, because I only have a couple minutes left, we had discovered that the glycolysis inhibitor, which is widely used both in vitro and in vivo, inhibited the growth of E cells only in macrophages, and this was true in vivo. We fiddled around with a lot of mechanisms and didn't find anything that worked. We looked at mTOR, we looked at autophagy, etc. What we did find, though, is that 2DG actually inhibited zinc acquisition in the histoplasma. And you can see here the survival of histo in the macrophage when we restored zinc into the yeast and incubated it with 2DG, you got no killing. If we did it with iron, you got killing. If you did it with magnesium, you got killing. Suggesting that unexpectedly deoxyglucose manipulated zinc levels. We don't know exactly how, but it looks like it has to do with import of zinc into the macrophage. So let me just conclude. Zinc is an important nutrient that supports intracellular fungal replication. GM-CSF is important in depriving the fungus of this metal through manipulation of zinc homeostasis. IL-4, in contrast, promotes survival of histo in the macrophage and one mechanism by which it does is by promoting zinc fortification of the fungus. And the unexpected finding of a glycolysis inhibitor depriving the organism of zinc was really a very interesting finding. So I'd like to thank a number of people. Mike Winders got us started on metallomics. Kavitha Subramanian, Vignesh continued their work. Julio Landero has been our chemist that we've worked with for many years. Logan Blanchett created the reporter. Diego Rossi did the work with 2-deoxyglucose. Alexei Parolo did all our bioinformatics, and thanks to Bruce Klein at Wisconsin, who collaborated, and Sanat Devanovich at Children's Hospital, who also collaborated. And with that, I will exit and stop sharing so Tamara can go.
George, thanks very much for a fantastic talk. And I'm going to hand the podium over to Jay now.
Thanks, Joe. This is Dr. Jay Coles from Tulane School of Medicine. It's my happy to introduce our second speaker, Dr. Tamara Doering, who did her undergraduate and graduate in medical training at Johns Hopkins, and her thesis was on studying GPI-anchored biosynthesis and trypanosomes, and then got into the mycology field with a postdoc with Randy Scheckman studying intracellular transport and Saccharomyces, and then turned to Cryptococcus after working briefly with Tor Castle. So Dr. Doering runs a lab at Washington University. It's well supported by a variety of funding sources, and really focused on mechanisms of synthesis of cryptococcal glycans, particularly capsule, cell wall, and other aspects of cryptococcal biology. And so Dr. Doering is going to talk on lipid transport in Cryptococcus neoformans.
Just get my pointer. Can you see the slide correctly?
Yeah, looks good.
Okay, great. Thank you, Jay, for that kind introduction. And I'm really happy to be here today to tell everyone some of the work that's going on in my lab about lipid transport in Cryptococcus neoformans. And I know most of you are familiar with this organism, which was perhaps originally a sort of a curiosity. It was this encapsulated yeast that was observed both in environmental sources and in the context of human infections. But I think now we think of it more as this World Health Organization priority fungal pathogen. It's in this critical group that's shown here and is also known for causing about 150,000 deaths worldwide each year. So just in case there's anyone tuning in who isn't familiar with the associated disease, it's called cryptococcus. And this begins when the infectious particles, spores, or perhaps desiccated cells are inhaled, and that sets up a pulmonary infection. And the resolution of this infection, as with many others, is going to be greatly dependent on the host immune status. So if individuals are immunocompetent, the infection is generally asymptomatic, although latent infection certainly occurs. And in contrast, if the individual is or becomes immunocompromised, you can get dissemination of cryptococcal cells really to many different tissues, but the really devastating outcome happens when it goes to the brain, and there it can cause a frequently fatal meningoencephalitis. So turning now to the organism, Cryptococcus, as you can see in the upper right, is a budding yeast in this thin section micrograph by Akioneta, former student. And just a couple of points about the organism that will be relevant to what I say later. We know that Cryptococcus can behave as a facultative intracellular pathogen, so it's pretty happy growing inside host cells or outside of them, and also out in the environment, as I've mentioned. And another feature of Cryptococcus is that it's distinguished by a polysaccharide capsule, which you can appreciate here in this thin section EM, where this is sort of the material that surrounds the cell wall, which you can see here. So my lab has really focused on the fundamental biology of this organism, but in a range of areas. So one thing we've been very interested in, some of you know for a long time, I've been fascinated in the capsule and how it is made. And just to show you the capsule a few different ways, here you see an India ink negative stain of cryptococcal cells, and this halo between the ink particles in this gray background and the edge of the cell here, the cell wall, is the space occupied by the capsule. And we also like to look at this in a little more detail. This is a quick freeze dip etch picture, again, of the edge of a cell. And what you see is these fibers sort of extending up and to the right from both the parent cell and a newly forming bud are the capsule fibers. So in the course of studying sort of in detail how capsule is synthesized, we've also gotten into working on other glycoconjugates. Particularly lately, we have quite an interest in the cell wall, which you can see here, the boundary between these capsule fibers and the plasma membrane over here, sort of double layered structure. So we're very interested in that. And we're also interested in how the capsule is regulated because it changes a great deal depending on the environment. In a sort of different direction, some other work that my lab is interested in has to do with really the physical interactions between the cryptococcal cells and host cells, such as phagocytes. And you can see that here where we have some green stained cryptococcal cells, and you can see the mammalian cells stained in red and blue. And actually thinking about these interactions has led us into the story that I'm mostly going to focus on, which has to do with today, which has to do with lipid trafficking. But I'll just also mention that other things we've been interested in over the last few years include how we can kind of assess the impact on virulence that naturally occurring variants in the genome have, and also features of fungal dissemination. But today, as I mentioned, I'm going to really focus on this sort of middle part of the slide, which has to do or starts off with looking at the interaction between these two different types of cells. So what do I mean by that? What I mean by that is that the cryptococcal cells shown here in green, we know that they can associate somehow with the surfaces of host cells and then be internalized by those cells. So that's sort of the beginning of a relationship, but the way that can then play out can happen in many different ways. And this, I should also say that these interactions in these early phases are very interesting to us because we like to think about the surface of these cells, the capsule, the cell walls, and the plasma membrane, which are going to sort of be what is likely involved in these processes. So in terms of how these reactions can go, of course, what the host cell probably would be happiest with is if the fungal cell can be killed, and that certainly happens in some instances. But we also know that there's a lot of other fates of these internalized cells. For example, work from the May lab and the Kasarval lab has established that the fungi can actually exit in a non-lytic way, this non-lytic exocytosis, apparently unscathed. And we also know another outcome can be that the cells continue to proliferate within a host phagocyte. And of course, this isn't always the best outcome for the host either, to have the cryptococcal cells sort of setting up residence inside. That can often lead to their demise as well. So just to sort of sum this up, what you're seeing is that there's many possible resolutions of these interactions between the host cell and the fungal cell. And really the balance of those different interactions is going to determine sort of the course of pathogenesis in the organism. So as I mentioned, we were quite interested in this early step for reasons I alluded to, and we decided to take a look at that. And the way we did so was to label the cryptococcal cells with Lucifer yellow. So that dye interacts with the cell wall of the fungus. And we then incubated them, well, we serum opsonized them first and then incubated them with mammalian cells for some period of time so that they could potentially be engulfed. And at the end of that time period, we could stain the host cell to be able to sort of see the different entities. So if you do that and take a confocal picture, you might see something like this. So here you can clearly see that some of the fungal cells seem to be associated near the surfaces of the host cells, where others are clearly internalized. And you can see that really nicely if you just look at the channel for the cell mask that's staining the host cytosol. Here you see these silhouettes of the yeast. So this told us we could potentially assay these interactions in this way, and other people have done some similar things. But luckily, we don't need to do that by confocal microscopy. Here's another image where you're just looking at fluorescence of cryptococcal cells, again stained green, and some slightly different staining of host cells. But again, when you look at eMERGE now, it's very clear that there's sort of a variety of different interactions that we can visualize in this way. And luckily, even quite some time ago, when we first were doing these assays, there was already software available to annotate images like this, which would allow us to quantitate, for example, cryptococcal cells that were perhaps inside or associated with host phagocytes. So with an assay like this in hand, and especially something that we can potentially quantitate, we decided to use this to do a screen. And what we screened was at that time a collection of about 1,200 deletion mutants that had been generated by Hita Madani's group at UCSF. And the question we were basically asking with these mutants was, which of them or which of these deletion mutants, which genes are important for this interaction such that the mutant might give us altered levels of uptake by the phagocytes sort of in either direction. So what you're looking at here is just relative uptake of the mutant compared to wild type. And you can see that we do get, sort of a number of hits in terms of mutations that clearly affect this process. And I thought I would just show you one of these, although a number of stories came out of this. And this was when we looked at a mutant that's lacking a gene called PFA4. And it turns out it's actually a palmitoyl transferase. It modifies proteins with lipids. And what was very interesting was then to try to track back, why would this particular defect lead to these terrible cell wall defects that you can see manifested here, both inside of host cells or even when the yeast are on their own. And I show this really to highlight the very beautiful work of Felipe Santiago Tirado when he was in my lab some time ago. So as I said, a number of interesting stories came out of hits from this screen, but we also thought that we could improve this. And a couple of things we thought could be improved about the way we were going about these screen. One was that in this initial set of mutants that we screened, we used cell lines. We used THP1 cells as a model phagocyte, but we were interested in sort of looking instead at primary cells, mouse and human. We also, as I mentioned, had taken advantage of having access to the partial deletion library that was available at that time. But in the intervening years, the Madani lab had greatly expanded this library about fourfold. So we wanted to look at the rest of it. And also our early assays had a little trouble distinguishing between the adherence of cells and the actual engulfment of cells. So we wanted to revise that. And I won't go through all the details here, but I'll tell you what is most important, which is who did this. And that was the work of Sydney Briner, who's a current graduate student in the lab, with working with Kuo Hua Chen, a former technician, as well as Ellie Gaylord, a former student. So when they went back and screened the now much larger library, so almost 4,700 mutants, what they found was that there were a few hundred of them that seemed to have defects in one way or another in interacting with either human or mouse phagocytes, primary cells. And there were about 100 of these that were common to both of those groups. So they looked at them a little bit further. And perhaps not surprisingly, if you look at what types of groups of genes are enriched for in the mutants that show these phenotypes, they're quite a range of very basic cellular processes. And again, it probably isn't too surprising because what we're looking at is these really big sort of interactions at the surface of the cell. Again, there's a lot of interesting things here, but I'm going to focus on one today. And that happens to be a protein that is involved in lipid transport, specifically sterol transport. And this is work that was initiated by Keely Choi, a former graduate student in the lab. And the current people working in this area in the lab are Dongyu Kim, who's a graduate student, and a postdoc, Cecilia Gutierrez Perez. So before I show you some of the actual data, I just want to go back and start with a little background to get everyone thinking in the same way about ergosterol. So ergosterol is sort of what fungi have in place of cholesterol in mammalian cells. And it's a sterol molecule. It's sort of, this is its structure at the upper right, and it's also cartooned in yellow here. So you can see it's this planar molecule with a small head group. It has some really interesting properties when it's incorporated in membranes because at low temperatures, the shape of this molecule actually disrupts the packing of acyl chains, so they don't form too rigid of a gel. And at a higher temperature, it actually serves to constrain the motion of these chains. And so in that way, it prevents the membrane from being too fluid. So the same molecule is sort of acting to buffer membrane fluidity in both directions. And this is really important because membranes need the proper composition and fluidity to perform all of their functions, whether that's cell division or motion or secretion, as well as the function of many proteins that are associated with membranes rely on this. So it's really key property. These kind of molecules are also involved in organizing lipid rafts, which are other important membrane domains. So we know in terms of fundamental biology, ergosterol is really important. And it's also really important in the context of antifungal therapy. And that's because it's really a key antifungal target, as many of you know. So specifically for Cryptococcus, there's only 3 agents that can be used to treat this infection. And the 2 of them that can be used as monotherapy, both are based around this particular molecule. So for example, amphotericin B, which is shown here, serves probably to extract ergosterol, which is shown here as these sort of maroon lozenge shapes, ovals, extract those from membranes, have been thought to have a role in pore formation. And then a second compound, fluconazole, is one of the azole drugs, and these are known to inhibit ergosterol synthesis by interfering with one of the early steps in the rather complex synthetic pathway. So, a little about the biology of ergosterol. It's actually synthesized in the endoplasmic reticulum, but it doesn't stay there. In the end, about 90% of it ends up in the plasma membrane. So, in a cell, about 90% of the ergosterol ends up in the plasma membrane. And in terms of concentrations, that's about 30 or 40 mole percent. That's sort of everything in the plasma membrane. And this compares to about 5 mole percent in the endoplasmic reticulum. So obviously the cells are generating and maintaining a really steep gradient here of concentration. So the way they do that is by moving the ergosterol around appropriately between the cells, both to establish that gradient and then to respond appropriately to environmental conditions that might require slightly different properties of membranes. And this movement of ergosterol, while primarily between the ER and the plasma membrane, can also involve the mitochondria and vacuoles. This movement can happen either vesicularly, so moving, for example, with the vesicles that are moving secretory cargo out from, sort of out toward the surface of the cell. But they can also happen in a non-vesicular way. And this has some advantages, probably in terms of the kinetics, that it can be a very rapid way to move lipids around without having to involve a whole secretory vesicle and proteins. And it can also serve organelles like the mitochondria that don't receive secretory traffic. So we know from model yeast, Saccharomyces cerevisiae, that there's a couple classes of these types of sort of lipid transporters and particularly sterile transporting molecules. And they're shown here. So there's some cytosolic molecules called Asch proteins that serve to exchange lipids for each other. So sort of pick up a lipid at one membrane and then drop it off in exchange for another. And you can see readily how that would sort of adjust membrane composition. And then there's another group of protein called the LAM proteins, and these are transmembrane proteins that occur at sort of areas of the cell where you have close apposition between different membranes. And they actually exchange lipid for water. So they're moving lipid, but without a second lipid usually involved. And this is the class that we're going to focus on today. So again, going to the model yeast, we know something about the LAM family proteins in that context. There's about half a dozen of these proteins, and you can see they're transmembrane proteins. They have these sterol associating domains, one or maybe two of those. And they occur in different areas of the cell where they seem to perform their functions. Interestingly, when you look at the cryptococcal sequence, there seems to be only one of these. It's the closest to the Cerevisiae YSP2, so it's named accordingly. And this was actually one of the hits that we got in the sort of overlap set when we did screens with both mouse and human primary cells, as showing aberrantly high uptake. And I should mention that this had also been identified a little bit earlier in a screen from the Laja and Donlin labs when they were interested in what mutants in the collection might have be particularly sensitive to echinocandidates. So YSP2 is supposed to be a retrograde transporter, so it's moving ergosterol from the plasma membrane into the interior of the cell, specifically back to the endoplasmic reticulum. And Keely Choi was interested in taking a look at this. So the first thing she did was sort of make a fresh mutant and a complemented strain so she could look at a few phenotypes. And what you see here, this is serial dilutions on the indicated media. And you can see that compared to the control cells, the mutant has quite some difficulty growing. I mean, it's fine in sort of rich medium conditions like YPD at 30 or 37 degrees. But if you subject it to some membrane stress like STS or amphotericin B, the mutant has quite some problems. And I should mention that it's not just the deletion mutant. If we also engineer a mutant where there's just a single site-specific mutation in the sterol binding site, you get exactly the same phenotype. And another phenotype we noticed was that the capsules on these cells were a little bit thin. And here you're looking at cells that have been induced to grow larger capsules by growth in what we call host-like conditions of 37 degrees, 5% CO2, and tissue culture media. So there's a bit of a capsule defect, as you can see quantitated below. And this kind of defect, combined with its sensitivity to membrane stress, suggested these wouldn't do very well in an animal model. And indeed, that's the case. Here you see mice infected inhalationally with these organisms, and the mice that got cells that were of wild type lose weight and succumb to the infection within a couple of weeks. We have to sacrifice them, whereas the ones that got the YSP2 deletion are totally fine for a couple of months. And that's reflected in the colony forming units that you can find in their lungs and brain, as shown on the right. There's really barely any in the mutant cells. So this supports the importance of sort of sterile homeostasis in these cells, certainly, and in the context of infection. But another thing that had interested us as we were looking at these cells was that they seem to have some morphological problems. So we're staining here with calcifluor white, which stains the cell wall. And you see that while wild type cells have this nice even wall structure around them, the YSP2 mutant has these thickened walls and that seem to be sort of protruding into the cell. We looked with a different cell wall stain just to confirm this observation. So here, Lucifer yellow again. And you can see again, the cell walls look very nice, smooth, even thin in the wild type and quite perturbed in the mutant. And this holds up also if you use a membrane stain here, philippin, that interacts with sterols. And so you see it's sort of going along with the same perturbed shape in the wild type cells, and then the mutant being quite perturbed. And I think you can also appreciate that this philippin staining is much brighter in the mutant, and that makes sense because this is a retrograde transporter, so it's bringing lipids back in from the surface. If it's absent, you get this accumulation that shows as the brighter staining. So we were very curious as to what these sort of structures actually were, and so we used electron microscopy to look at that. Here you're looking at wild type cells with a nice even cell wall and just a little bit of capsule. They're grown in rich medium here, so they don't make much. In contrast, if you grow them in the host-like conditions, now you see a much thicker cell wall and a quite more dense and elaborate capsule. That's wild type. If we look at the mutant in rich medium conditions, it looks pretty much the same, no real difference there. But if we now stress it under host-like conditions, you see there's tremendous aberration in the cell surface. So first of all, you have a smaller or thinner capsule, as I pointed out earlier. And while you're getting the thickening of the cell wall, you're also getting this sort of invagination of material. And it doesn't seem to be just the cell wall, but also membranes that go along with it. So we were trying to think about this and figure out what's going on. And one question was, is this a cell wall problem that's kind of collapsing into the cell and bringing membrane along with it? Or maybe is it the other way around? It's a membrane problem that's sort of causing the cell wall to be deformed. And where was this material coming from anyway? Is this sort of, again, collapsing in or being synthesized anew from the start? So to answer these questions, we decided to take, especially the second one, we decided to take a pulse chase approach. So here we stained cells with Lucifer yellow at the beginning of an incubation in host-like conditions, and then came back a day later with the second cell wall stain. And at the end of that period, if you look at the cells, you see that the initial stain is still looking fine. It still looks, like unperturbed cells. But if you look now at the cell, at the calcifluor white, the second stain, you see, of course, the outside is stained again, but now you're also sort of revealing all of this material, which must have sort of been generating, it's new. It was made in the course of this incubation. Here's just the merge. So based on the data I've shown you and a few other experiments, we came up with a model for what we thought was going on, and that's shown here. So the idea is that YSP2 is a transporter that's involved in bringing ergosterol back from the plasma membrane to the ER. If it's missing, excuse me, if it's missing, you now get an accumulation of ergosterol at the membranes, and we think that this perturbs the fluidity of the membranes such that they start invaginating inwards. Now, it's just not just the membranes that are sort of going to now come into the cell. It's also going to be proteins that are sort of housed within that membrane. And many of those are involved in making the cell wall. And that's why we think that as the cell sort of continues to grow, even in this perturbed state, you start to get this new material made inside of these invaginations. We have a little support for this idea, and that's by looking at a plasma membrane protein. This is actually plasma membrane ATPase, which is not involved in making the cell wall, but it is in the plasma membrane. It's tagged here with M neon green, and you're looking at a maximum intensity projection, which I think shows quite nicely that this protein is sort of present all over the surface of these cells. In wild type cells. But if you now look at how it behaves in YSP2 cells grown in these conditions, you start to see these aggregations, accumulations of material that look like they're sort of coming in from the surface of the cell. And these puncta are just quantitated at the right. They're very unusual in a wild type cell. So that supports this model. Another thing, of course, that a model can do for you is make predictions. So one thing that we could predict from this model is that all of these sort of bad downstream outcomes could be avoided or at least mitigated if we could reduce the flow of ergosterol to the plasma membrane at this step in the mutant. And I've already mentioned a way that we can potentially do that, and that's by using azole compounds to inhibit the biosynthesis of ergosterol. So what we did was to grow cells in different amounts of fluconazole. And here you see cells that have been grown for a day in no fluconazole. And our wild type cells are doing fine. The mutant is doing pretty terrible. But if you start adding more fluconazole, what you see is these cells, in fact, start to grow better in the presence of drugs. The mutants are dramatically improved. This isn't resistance, because if you keep raising the concentrations of fluconazole for this incubation, or this culture period, then what you see is that all of the cells die with sort of equal kinetics. And this is because they're accumulating these toxic intermediates that are the intermediates that are formed under the azole inhibition. But what's interesting is it's not just in this part of the curve, it's not just that the cells are growing better, they actually reverse all of this or most of this aberrant surface structure that we had observed with the same kinetics. So I think this supports our model well for what YSP2 is doing in Cryptococcus, but of course it leaves some other questions. So one of these questions is, what is going on with the capsule alteration? My lab has shown many years ago with Amy Reese that capsule interacts specifically with elements of the cell wall. So it could just be the sort of disruption of the cell wall that is perturbing the capsule. And we, in fact, know that its architecture is perturbed in these cells. It also could be the fact that by having too much ergosterol on the outside of the cell, you're functionally depleting the endoplasmic reticulum and therefore probably perturbing its function as well, which may shown in terms of, sort of may manifest itself in perturbed protein secretion or protein function. And we also know from work, for example, from the Cronstad lab, that problems with ER function can certainly lead to capsule alterations. So that's one avenue that we still have to explore. And another avenue, coming back to lipid transport, has to do with why cryptococcal cells seem to have so few transporters. I showed you that Saccharomyces cerevisiae has half a dozen, for example, of these LAM proteins, but crypto only seems to have one. And the OSH proteins, which I didn't talk about, the cryptococcal complement of those is also a bit lower than some other organisms. So one possibility is that we're just not recognizing other ones, that maybe they're in the genome, but somewhat different proteins are doing these jobs, which would be one interesting possibility. And another interesting possibility is that perhaps this YSP2 protein is just doing more than it does in other cells. I mentioned in Saccharomyces cerevisiae, it's working at the plasma membrane ER contact sites to sort of adjust cholesterol or sterol concentration. But what we notice in Cryptococcus when we now tag this protein with the M neon green is that it's present not only at the cell surface here, but we also see some inside the cell. So it's possible that it's acting at other sites, which of course raises other questions of, how does it help maintain the specific compositions of those organelles, which are all distinct. So I'll just take a few last slides to tell you how we're going about trying to really approach this second question, which is finding more about or learning more about the machinery by which Cryptococcus moves lipids around the cells. And these experiments started with some observations when we were doing these sort of testing the phenotypes of the wild type and the two mutants that I've mentioned by serial dilution on various stress plates. And what I showed you already is that the two mutants don't do very well under conditions of SDS stress, but we noticed that actually there were a couple of colonies coming up that seemed to be growing quite well. And we speculated that these were suppressor mutations, which have acquired some genomic change that allows them to grow even in the absence of the YSP2 transporter, presumably because these changes have some direct or indirect effect on sterol transport. So Dong-Yu Kim in the lab sort of has taken this on, and he's now got in hand several hundred candidate suppressors, or he isolated that many. And about 40% of those were actually stable after further passaging and testing. And when he looks at the whole genome sequence of, well, he's looked at about 91 of these so far, he certainly sees non-coding mutations, and those could be interesting, but we have not pursued them yet. But he also has identified about 40 mutations that would be predicted to have high to moderate impact on the encoded protein sequence. And these coding sequences are in about 29 genes. So thinking about what you might expect to get out of a screen like this, given the data that I've already showed you about the fact that the azole drugs can help these mutant cells grow, what you would expect is that mutants perhaps in different points of the ergosterol synthetic pathway would turn up in this screen. And for that reason, we were quite pleased to see that a number of the genomic changes he identified were actually in these genes or in regulators of sterol transport. So that really nicely validates the screen and has encouraged us to go looking at other hits in the screens, especially some novel proteins. And we're pursuing this because we're really interested in sterol transport. And of course, this is sort of intimately associated with other kinds of lipid transport and phosphoinositide transport within the cell. And what we're doing right now is sort of taking some biochemical approaches to express domains of these proteins of interest, particularly the unknown ones, and so that we can look at what these domains do in terms of biochemical activities and binding characteristics, and maybe potentially whether they bind to sterol-related small molecules. So hopefully another time I will be able to give you an update on this. And meanwhile, I will just go forward and acknowledge the terrific members of my lab. So on the left, the names and the pictures are current lab members. And on the right, I have to acknowledge terrific students, two of whom were involved in the work I've shown you, Keely Choi and Ellie Gaylord, and also Liza Loza, who has worked on cell wall in the past, although I didn't have time to present that work. And I also would like to acknowledge our collaborators. So Michael Brent works with us on computational biology. Wandi Beatty made those beautiful, most of the EMs that you saw. And we're also working with Janice Anderson and Luca Laraya in terms of looking at the chemistry and biochemistry of these lipid transporters in Cryptococcus. And now I guess like the others, I would be happy to take questions, or like George. So I will stop the share.
Fantastic, Tamara. Thanks so much. All right, we're going to launch into the question and answer period. And George, could we ask you to turn on your camera and unmute? And I'm going to combine the first two questions because they have a related theme, George. And it centers around the question of a lot of us took a multivitamin this morning that also has minerals in it, including a certain amount of zinc. And the question is that a good thing or a bad thing, or is there enough buffering so that it's neutral? And then for people who might be susceptible to histoplasma, does that advice change in any way?
So zinc therapy is useful for diarrheal diseases in kids. That's clinically proven. The data with colds that people took is iffy at best. There's a lot of yeses and nos. The problem is that zinc, like many other things we ingest, is highly regulated by importers and exporters. So actually there are 24 zinc importers and transporters, which is really pretty amazing when you think about it from an evolutionary point of view. So even if you take more, there's a saturation point and the rest is going to go out. Um, so, so in terms of disease states, there's no evidence that zinc would improve outcomes with histo. You want, in fact, you want less zinc inside the macrophage to deprive it, and that would be true for a number of organisms. There's also, and we haven't seen it in histo, but there also is a process of zinc intoxication that has been proven in TB and a couple of other organisms. But again, if you take a lot of zinc, the body will accept what it needs, and the rest is useless.
Okay, I'm going to hand it over to Jay to continue with questions for Tamara.
Okay. Thanks, Joe. Tamara, one of the questions in the Q&A is there a way to separate the engulfed Cryptococcus from the Cryptococcus that's still maintaining outside the cell? And if so, are there structural changes or lipid or polysaccharide changes in engulfed organisms? And I guess that makes a difference, I guess, where it is, right, in the early endosome versus maybe an endosome that's fused with a lysosome, phagolysosome structure.
Yeah, thanks for the question. So we can certainly separate those visually, as I think I've shown you. As far as physically, I don't think we've ever tried. I mean, in these experiments after this engulfment interval, we watch the cells, certainly. And I guess we could, we probably could try to find a way to release the ones from the outside if we wanted to look at those separately. As far as the ones inside, I think, I would expect they're almost certainly changing because they certainly respond to the host environment. And even if you just look at cells once they're engulfed, they become very heterogeneous. The capsule change, the cell walls change. I think the trick would be recovering them and to the point where you could sort of get good data about the changes. So it might be possible. I think it would be technically challenging.
Thank you.
Okay, George, another question. This one's from Chad Rapley. In your isotope labeling experiments, it looks like the yeast are mainly acquiring the 68 isotope of zinc instead of intracellular zinc pools. And so the question is there something special about MT3 and the pathway that's going, directly from outside the cell to the yeast in the phagosome and bypassing cytoplasmic pools of zinc? How does that work?
Well, the MT3 would be picked up. The MT3 would pick up zinc in the cytosol and then end up in a lysosome or phagosome. And under the right, if the cathepsins are activated, which would be true with IL-4, they would release zinc, which would then could exit through a number of exporters into back into the cytosol and then into the phagosome where histo could take it up. So MT3 actually, as I mentioned, behaves opposite the way MT1 and 2. They're very stingy. They will bind 7 zincs and only give up 1. MT3 is, the generous donor and will give it up under certain conditions, and particularly with cathepsin activation of the MT3. I hope that answers this question.
I hope so too.
If not,
Okay,
you can
Jay.
email me.
Yes, so Tamara, there was a lot of interest in the in vivo mutant work too, and some questions around that. So, so presumably the inoculum is the same, right? So at early stages of the infection, the fungal burden must be similar. So what's the fate of the YSP2 mutant? Is it able to attach or do any growth in vivo? And is there any kind of adaptive immune response to the mutant in terms of either a T cell or humoral response, and therefore could the YSP2 mutant potentially be like a vaccine type strain?
Right. So for the real answer to the immune response and whether this could be a vaccine response, I would have to get some of my immunologically oriented colleagues who are on this call to help out. So we haven't looked at that. In terms of the fate of the cells, I'm trying to remember. I think we looked pretty some earlier time points in infection. I only showed the burden at the end. And as I recall, those cells are cleared out pretty quickly, which is not surprising. They're very stress sensitive, especially at higher temperatures. So I think they're cleared out fairly rapidly. So that's sort of the answer to that part. I think Keely was always upset there was like one colony that she didn't have quite zero on that plot at the end. But in general, I think they're cleared out pretty fast, and I don't know about the immune responses. So happy if someone wants to look at that with us.
Great. Thank you.
Okay, George, there's a question from Bruce Klein. What are the driving forces for histoplasma evolution of the yeast zinc acquisition strategies for surviving in humans when most of its natural life is in the mold form in the environment? Or is it natural infection of bats presumed to be a possible driving force for yeast zinc acquisition?
So it goes well in nature, and we've actually looked this up. There's quite a bit of zinc in the soil, so it is adapted to a higher zinc content in its environment. In fact, the zinc content was actually from a study at the in Wisconsin. Going into the macrophage, the amount of zinc is considerably less. The amount of available zinc is in the picomolar range. So histo has at least 2 importers of zinc, like other fungi, ZIRT1 and ZIRT2, and they're turned on when zinc is low. So the driving force to acquire zinc is really driven by the intracellular environment as it goes from the mold to the yeast, since it's going from a rich zinc environment to a very low zinc environment. Now, whether that evolution occurred in bats or in, say, amoeba in the soil, I don't know the answer. I don't know the answer to that question, but obviously, obviously it's evolved over time, and it doesn't even have to do with humans. It could be small animal rodents who get infected, like mice in nature.
George, I know that you find histoplasma associated with bat guano in caves, but I haven't heard that the bats themselves get infected. What's the story there? Do bats get infected with
They don't get
histoplasma?
sick.
They get infected, but they don't show
Yeah, they
symptoms.
shed it.
Ah, okay. So that's very different than the association of Cryptococcus with pigeon guano, where it just comes there after the fact. It's not shed by the pigeon.
Right. No, bats will shed it.
Okay. I didn't know that. I learned something. All right, Jay, over to you.
Thanks, Joe. I'm going to combine 2 questions because so we can fit them in before the break. So Sean Duffy asked about the YSP2 mutant, whether it's tied to TORP2, and does the YSP2 have any altered rapamycin or torn sensitivity?
So we haven't actually looked at that, but I would not be surprised since, in fact, probably this YSP2 has an even more dominant role in terms of overall lipid traffic, I suspect, overall lipid traffic in these cells. So I would not be surprised about that, but we didn't look directly.
And then the follow-up question is about whether there's extracellular vesicle formation in wild types versus knockouts, and if the YSP2 mutant secretes less EVs.
Yeah, that's an interesting question. I could almost imagine it going either way. I mean, what's happening to membranes coming coming into the cell isn't really a normal sort of endocytic type of process, but it is, pulling a lot of lipids into the cell. On the other hand, these cells are just kind of sick. So often in those circumstances, you'll get more release of membranes in various forms, whether they're sort of actually intact, sort of intentional, for lack of a better word, extracellular vesicles is hard to say. And I think I would actually, that's kind of the problem. I mean, we could certainly look at that, and maybe I should ask Marcia to take a look at the strain. I can't remember if it's one of the ones we sent to him. But I do worry that these cells, especially under the host-like conditions where they start to show these perturbations, are just not healthy cells. So, I always worry about what kind of conclusions you can make when the cell is just overall not functioning very well. We'd have to think about how to do that.
Yeah, good point. Thank you.
George, another zinc aficionado in medical mycology is Duncan Wilson from the MRC Center in Exeter. And he has these really interesting studies that he published recently about the zincophore, PRA1, that Candida albicans secretes. And then that is a PAMP that leads to neutrophil infiltration into tissues. And if I understand it right, histoplasma might have lost PRA1. Um, could that have been part of the evolutionary pathway to adapt to survive and cause animal infections?
Yeah, Doug and I have actually discussed this, and if I remember correctly, and I don't want to put words in his mouth, but he thinks that the soil environment is one of the main drivers of having the zincophore in terms of the dimorphic. So blasto actually has the zincophore, and the environment is different in terms of acidity than it is where histo resides. And so that could actually, so histo doesn't have that zincophore to capture, but it does a good job of capturing zinc independent of that zincophore. But it could be very important I would agree, it might be very important in the pathogenesis of the infection and its adaptation to mammalian tissues.
Mm-hmm. And there's a number of species of histoplasma that have been defined over the last couple of years and named. Daniel Matute
Yep.
has been driving a lot of that. And I don't know if anyone's looked across all the histo species to see if they all lost PRA1, but it would seem like that would be an interesting question.
Yeah, I think so too. There's Mississippi and Ohio NCs, so I don't know how different those soils are between those two regions, but I think it would be useful to see whether just the zinc importers and exporters, the putative importers and exporters in histo, aside from just the zincophore, might differ amongst the various strains. And the other thing is, and maybe it's too soon to do, is as this organism moves west, it adapts to a new environment. And will
Mm.
that be an evolutionary pressure for it to somehow change its, at least, zinc acquisition properties? And I don't know.
Mhm.
It will take years to find out.
Daniel Matute was just here last week, which is partly why I've got this on my mind.
Yes,
But he's got all these genomes now, and he's also
right.
starting to do a lot of population level genomics. So, whole genome sequence for multiple isolates. And this seems like the perfect case where you two could team up and really go to town to look at
I'll
presence and
con
absence.
if he's not listening, I'll contact him.
Okay, great.
If he's listening, I'll still contact.
Okay, sounds good. Jay, I know there's some more questions in the chat for tomorrow, so
Yeah.
over to you.
Yeah. So actually, this one just came in from William Chirico about have you been able to do any live cell imaging? Because you showed the cell wall looks pretty normal in the high nutrient conditions, right? And then like if you start to change those conditions, um, can you actually see the invagination, or is there also maybe some so kind of more like an endocytic effect versus ability to reconstitute the cell wall, I guess, through a secretory process?
Right. So we have not sort of tried to look at those in terms of really live cell imaging and watching these events happening. We have looked at some different time points of the development of these imaginations, which is obviously a very different thing. But I think based on what we think is going on in terms of where the main defect is, it's really at the plasma membrane with the accumulation of sterols. So my bias would be that what's happening is that that's the perturbed membrane and that's the problem. But I guess we can't really rule out. I'm sure there are also problems with sort of vesicles fusing with the surface in that same time frame because, of course, the ER is, as I mentioned, getting a little bit starved for the same lipids because they're accumulating at the surface. And that gets a little complicated. It's actually, if I can jump onto, this sort of relates to another question that I see that's been asked, which is about how the sterol localization or amounts of lipids are changing in these mutants. And for the same reasons, this gets complicated. You've got an accumulation at one membrane, the plasma membrane, and you've got essentially depletion in the endoplasmic reticulum. And it gets a little complicated. I didn't show these data, but if you start looking at what's happening in terms of the regulation of the genes that are required to make ergosterol, there are some changes in terms of, the cell appears to be trying to increase its levels of ergosterol, even where the problem is probably that it has too much, so there's these complicated feedback loops. So that's in terms of, what's going on in the cell. In terms of how we can look at that, there's a number of ways that we've tried or can try. So we've done some composition, and it does seem like there's an overall increase in the amount of ergosterol that's sort of subtle. We still need to tease out sort of intermediates in that pathway. We can also use the Philippin staining a little bit. That's hard, works best at the surface where the levels are higher. We've also tried expressing domains of proteins that are supposed to bind sterols. That has not worked out super well for us either. But there are still some other ways that you can try to get cells to take up fluorescent lipids. That requires a little bit of strain engineering that we're working on now. So long answer. Sorry about that. We're sort of trying to get at all this, but we're a little bit still in the methodological weeds on that.
And George showed a kind of surprising effect of 2-deoxyglucose in the histo model. So what are the energetic requirements for YSP2 function in terms of glycolysis versus where's ATP play a role in this process?
Yeah, we haven't. It's a good question. We haven't really thought about or looked at any of the energetics of this.
Yeah. Thank you.
Tamara, there's another question that came in the chat that I'm going to let Jay ask you, but I thought I have a question for you. In your YSP2 mutant, part of the model is that you've got a big excess now of ergosterol in the membrane. And I noticed you found those mutants are hypersensitive to amphotericin B. And there's two models for how amphotericin B works. One is a sponge and one is a pore. And if you had more ergosterol, you might think you'd need more of the sponge to be toxic. So I'm wondering if you could think that part through. And then I'm wondering if Marty Burke's amphotericin B analog that only has sponge function but not pore function might be another tool in your toolkit to get at what's going on in the cell membrane there.
Yeah, it's an interesting question. And I have to say, the first couple of experiments when we did showing that amphotericin B sensitivity of the mutant, it's sort of counterintuitive, like, wait, there's more. Shouldn't that help the cells? But it's also seen in cerevisiae as well with the corresponding mutant. So and I think the way I think about it is that probably you'd have to assume like that the drug is not actually limiting, and more of the membrane occupied with this lipid is sort of more that can be damaged. But I agree, thinking about the sort of sponge model versus pores, and it seems like sponge idea has sort of gained traction over the last years. It is a little bit puzzling, and that's really interesting idea. Had not thought about that to try looking at that analog. So thanks for mentioning that.
Great. Yeah, so this question came in from Mike Sukai about so apparently oomycetes are treated with a sublethal dose of a drug, oxythiopropolin, right, an oxysterol binding protein inhibitor. And then during treatment, there's a massive accumulation of these intercellular vesicles that occur. And so, in relation to the YSP2 function, is there also importance for these oxysterol binding proteins in the crypto?
Yeah, it's a great question. I mean, these are a super important set of proteins, very relevant to this topic. And we're actually looking at those. They are a little more complicated. Cryptococcus or neoformans, and we have several of them. And that's some of the work that Cecilia Gutierrez Perez is doing together with an undergraduate, Stephen Klein. And so we are looking at those, and I think the question disappeared, so I can't see all the details of it anymore. But that's certainly going to impact what's happening in the YSP2, and we're looking at those. These are really interesting proteins. They're localized in different parts of the cell, and they don't all handle sterol. This gets at this idea that, it's like a spider web. You pull one strand, everything is going to move around, which is what happens with metabolism and certainly with lipids. Like, you can't talk about sterols in isolation because sometimes there's some kind of antiport with sterols in exchange for some other phospholipid or phosphoinositide, so I think these things are all related, and there's very little known in the system. So we're really looking forward to working on it some more, trying to sort it out.
And genomically, are there differences between Saccharomyces cerevisiae and these oxysterol binding proteins, putative?
Yeah, I mean, there's fewer, but of course that, in cerevisiae you have the genome duplication, but there seem to be several in crypto that look like those. And then there's another one that looks a lot more like a POMB ash protein.
Yeah.
So we're just currently engineering the strains and getting ready to look at those in more detail. Thanks for the question.
Sure.
Well, terrific. We've gone through all the questions that were asked, and we're right near our ending point. So I want to thank again our two fantastic speakers, George Deepe and Tamara Doering, my co-host Jay Coles. And I don't know about the rest of you, but I'm about to jump off and then find the YouTube video for Johnny Cash, Beans for Breakfast, and listen to that song.
You can't miss it. Johnny, as you guys know, Johnny Cash is great. And that song is a true country song.
I don't think I've ever heard it.
Yeah,
I know it's not, it's not one of his more common ones, but it's a great one.
Well, thanks for the recommendation, and thanks to both of you for sharing your wisdom and insights with us. Fantastic.
Thanks for the invitation. Really fun.
Yep. Bye.
Thanks.
Bye.
Bye,
Bye,
all.
Jay.